Thanks for joining us for this symposium. I'm Bill Rapisi, President and CEO of the Lymphatic Education Research Network. LE&RN's mission is to fight lymphatic diseases through education, research, and advocacy. In order to win a fight, you first have to join it. So we ask, please become a supporting member of LE&RN at lymphaticnetwork.org. And we hope you enjoy today's symposium.
All right. Hello, everyone. Welcome to the Lymphatic Education and Research Network online symposium. My name is Isaac Lee, an assistant professor of Cornell University, and I'm excited to share some of our study on bioengineered models for lymphatic research. All right. So so we developed some of these tissue engineered models for lymphatics, and I started my lab around 5 years ago at Cornell Biomedical Engineering, and I'm very happy with this opportunity to share some of our study, and potentially this may inform some of these exciting results. Also have some opportunity for future collaboration as well. I'm from Ithaca, so Cornell is located in Ithaca, and Ithaca has lots of gorges and water. So I just had some of these photos like Taganrog Falls and Ithaca Falls and Buttermilk Falls, and we had great time this summer here. And now, outside it's great weather. And Cornell is located here besides the Cayuga Lake. And with this great nature, my family is growing with now 4 boys. And with my wife, currently as a nurse, she works for patients in the hospital, and I work in a lab, both of us working for human health. Also, my lab, since joining in 2019, is also growing, and I'm very, very happy with all these talented young people in my lab. And also, I must acknowledge this Lymphatic Education and Research Network Postdoctoral Fellowship. I think 8 years ago, I got funded 2016 to 18. At that time, I was working something different project, but this funding really helped me to focus on lymphedema, and that funding and that study still impact in my work at Cornell. So I really appreciate their generous funding and support. All right. So my lab is working on lymphatic with some interesting model system called organ-on-chips. So we use this chip system to culture human lymphatic cells and with other cells in vitro 3D to mimic some of these lymphatic behavior, like lymphatic sprouting and cancer interaction with lymphatics, also lymphatic drainage, and some of the morphogenesis of lymphatic valves, and interaction with lymphatics and with immune cells in different contexts. With this model system, we study some of the lymphatic related disease by adding some environmental factors or genetic factors and some other factors to model the disease, and then understand the mechanisms of this disease and propose potentially new therapeutic strategies for these different types of disease. So with this approach, My lab focused on three different types of human disease, but these are all lymphatic related context. The first is lymphedema, the swelling of arms and legs due to the lymphatic dysfunction. The second is cancer. So we study pancreatic cancer and breast cancer, working on the metastases and cancer immunotherapy. And lastly, we study neurological diseases in the brains and in the eyes. So today, due to the limited time, I would more focus on the lymphedema and some of the neurological diseases. So I guess many of you in this symposium know the lymphatics and their roles in human biology. But if you're not familiar with that, I always bring this type of comparison with these lymphatic vascular system with this sewer on the street. After heavy rain, we need these water on a street to be cleared, and the sewer on the street serve as a draining system. Similarly, in our body, we have vascular system with artery and vein, and there might be some leakage in between these arteries and veins, and this leak fluid should be cleared away by another type of vasculatures, which is lymphatic vessels. And this drain fluid filtered in lymph node, and they get into this returning to this vein through these subclavian veins. So it is recirculating all this fluid, maintaining the fluid homeostasis. If we have issue in this lymphatic system, as you can see this photo, the right up, you will have this flooding on the street. And similarly, in your body, you will have this abnormal buildup of this interstitial fluid that can make this abnormal swelling of these arms and legs, which is lymphedema. And there are more than 150 million lymphedema patients in the world, but still we don't have clear therapeutic approach to treat this disease. And usually when we see the doctor, we got recommendation having these physical therapies like massage or wearing some compression garments to physically propel this fluid from this swelling area. So my lab now focused on this lymphatic disease using this engineering tool. And I just brought a schematic of lymphatic vascular system, and it has quite complex structure, but largely has two different types of vasculatures in their network. One is initial lymphatic vessels or lymphatic capillary, and the other is collecting lymphatics. And this initial lymphatic, as being called initial, they initially uptake this fluid from this interstitial space. And these initial lymphatic is rarely covered by basement membrane or other mural cells, and also have some highly permeable button-like lymphatic junctions. And this fluid get into this lymphatics through these lymphatic vessels. So the main hallmark functional property of this initial lymphatic is button-like lymphatic junctions to allow fluid drainage into this initial lymphatics. But once this lymph formed in the lymphatic system, this lymph should be transported to the lymph node without leakage or without backflow. To achieve this collecting lymphatic has a much tighter junction called a zipper-like junction. Also, it has interluminal valve to prevent backflow. Also, it is covered with some muscle cells And these lymphatic muscles really make this motion of this pumping, propel this lymph to the lymph node. So zipper junction in collecting allows lymph fluid, lymph flow within the system without leaks. So now we focus on these two different types of junctions in the device and how we could understand lymphedema. And we know this lymphedema has many different causal factors, but secondary lymphedema or acquired lymphedema, they cover the majority of these patients compared to the genetic primary. And this lymphedema, secondary lymphedema, also because many different reasons like surgery of lymphatic system, mostly in breast cancer, but many times it is post-infectious and inflammation related. So within that, with this fact, we wanted to build up a lymphatic system in vitro and study some of these mechanism of this secondary lymphedema under inflammation. So the lymphatics on a chip, they look like this, and it has their size is this size, almost 1 centimeter and 2 centimeter dimension, and this is the top view of the device, and the white part is PDMS, is a type of polymer, which is transparent, not toxic to the cells, and this polymer is used to make the device basically. But this PDMS is highly stiff, so it is like a rubber. So cells, they are not happy when they directly contact PDMS. So we include within this PDMS device type of hydrogel. It is collagen 1 from red tail, and this hydrogel is introduced within this device. If you see this cross-sectional view, if I cut this part here and see their cross-section view, you can see the device in white color. And within the device, we have another empty space, and this space filled with collagen 1. And within the collagen 1 space, we have two empty hollow channels. To make these two channels, we insert a microneedle through these two channel through this kind of structure, and then pour collagen. So collagen is initially liquid, and they can become solid at high temperature, and then they are filling this area with this needle, and they become solid. And then we remove these two needles, and that leave these two empty hollow channels. And this channel is completely empty, at the same time completely embedded in collagen 1. That means if you seed lymphatic endothelial cells into this hollow channel through this reservoir, these cells cannot hear the collagen directly, forming a tube-like structure in 3D collagen. And then we introduce flow through this reservoir into this lumen. And then these endothelial cells, they are initially single cells, but under the shear stress, they form some level of junctions and make some monolayer of the lymphatic structure. So this is the vessels in 3D and the cross-section view of the channel. Clearly, it has some lumen. but the other part of this channel is empty, and we add some lymph molecules like fluid and some other particles. With this system, we wanted to recapitulate some of these lymphatic specific junctions, and the button junctions are the key hallmark of this initial lymphatics. And there have been many studies from many lymphatic researchers using different model system. And now from Dr. Melody Swartz studies this interstitial fluid and making this lymphatic junction be more permeable on the transwell system. And some other models also showed its things. And we wanted to little more investigate more than that to compare different types of flow, like luminal flow versus interstitial flow, or both flow in different stimuli like VEGF-A or VEGF-C conditions. So to do that, we using the same device, we put them on the rocker, and this rocking system allow them to have luminal flow like this direction, so through this direction. And it is not unidirectional. They are oscillative, but very slow, so they are not turbulent. So they are luminal, so laminar flow. And at the same time, we introduce some high amount of volume in one channel through these two reservoirs than the other channel. So these high difference can cause another types of flow, that is which is interstitial flow. So we could have 4 different flow types. First, no flow and luminal flow only, and interstitial flow and both flow in VEGF-A and VEGF-C condition. And we compare their junction morphogenesis in this device in different flow condition. We found that the VEGF-C plus interstitial flow show very, very high amounts of button-like lymphatic junction compared to other condition. But one very interesting thing is that when we examine the lymphatic sprouting, these VEGF-C condition, they didn't show that much sprout in any types of flow, but VEGF-A show very much sprouting under interstitial or interstitial plus luminal flow. So we thought these flow and these different types of growth factor make some different outcomes in lymphatic junctions and the sprouts. So now we concluded like this. So interstitial flow plus VEGF-C, they cause button junctions in LEC channel, but without sprouting. Interestingly, the same flow plus VEGF-A, they show highly zipper and with sprouts. So still we are not clear the mechanism in beyond this observation, but we now understand interstitial flow plus VEGF-C would be one of the important conditions in forming this initial lymphatic button junctions. In other separate study, we also studied the role of ECM, extracellular matrices, in lymphatic junctions and barrier. And we found integrin alpha-5 play a role in this junction formation in lymphatics. So in the same device, initially we use collagen 1 in this chip, but we just added a little bit of fibronectin. I remember this is 50 microgram per ml in solution, and they were just added to collagen gel. And these lymphatic channels shows quite different junctions. So collagen 1 show this diffuse junction, but this is affecting. This is not a VE-CAT, but it tells some cell structure. But if you have some fibronectin, they have these very tightened junctions. And interestingly, integrin alpha-5 are highly activated in this condition. It is known because fibronectin is the ligand for this integrin alpha-5, but collagen 1 with LEC, they are much less activated. So in the next experiment, we didn't use any fibronectin, but these are just collagen 1. But we artificially activated integrin alpha-5 using a type of antibody of alpha-5, which is SNAC51. And this antibody can activate integrin alpha-5 without ligand. So they serve as a kind of pseudo ligand. And once we activate alpha-5 integrin in lymphatics in collagen with no fibronectin, they also show similar zippering. With this quantification, they have higher zipper in the junction area. And we added some of these dye, like I think this is dextran. And this dextran is labeled with some fluorescent dyes like FISI or Alexa something, and they are imaged in real time in 0 minute and 1 minute. And control lymphatic has highly leaky, but this integrin alpha-5 activated lymphatic show much better barrier functions, as shown in this permeability is reduced. This study received Microcirculatory Society Award for Excellence in Lymphatic Research 2021. I really appreciate this recognition from the society. With that, we could develop this bona fide button junctions in lymphatics on a chip, and we compare this LEC with BEC. In different types of antibodies. And one of the beauty of this device is that we can fix them and stain them and observe their structures in details in 3D manner. So if you see the BEC, BEC is blood endothelial cells, see this VE-CAT adherent junctions is highly colocalized with CD31, and they are they are continuous like this. But in LEC, they look quite different. And I had two different types of arrowhead here, red color and yellow color. And this red color means this part, VE-CAT is positive, but CD31 is negative. For example, here, this is VE-CAT positive and CD31 negative. By contrast, this is CD31 positive and VE-CAT negative. So overall, they show some of these interdigitated junction formation, which is known since 2007 by Peter Barlook and McDonald Labs, discovery of this button junction in mouse lung models. So we were excited about this discovery and study some of these lymphedema and lymphatic drainage by this schematic. So we have lymphatic here. This is mimicking initial lymphatic with button junctions. And if you see this image here, this is the device in the incubator. And the right side channel has lymphatic, and the left side channel is empty, acellular channels, which is filled with high volume fluid like this. So can have this pressure gradient, can cause convection from left to the right. It is mimicking this interstitial pressure in sub-Q area. So, and to do that, we added some fluid or including some particles, insoluble microparticles or fatty acid with different size of carbon chains. and phospholipids and albumin. And all these different molecules are labeled with fluorescent dye, so we could measure these drainage by measuring their volume, also measuring their intensity. So by getting that, we can calculate the amount of these drain components and then also remaining parts. So we can calculate the lymphatic drainage in vitro. So we compare this LEC with PEC in the same device, same cell seating condition. And this LEC show much better draining ability of these different types of lymph molecules compared to PEC. Clearly, no cell barrier is the easiest things that drain these things much quickly. So we set this no cell as 100% drainage, and compared to this control, lymphatic shows almost around 60% drainage in different types of molecules, but PEC show around 20% or less than. And we image them, and this is one of the representative image of cis-16 fatty acid in red. Upper one is LEC in 1 hour and 2 hour, and this cis-16 really entered this lymphatic quickly, but PEC was much less. And this lymphatic lumen, this blood endothelial lumens is mostly dark, which means there's no red color dye compared to this LEC, which is really mimicking the nature. So we know this lymphatic drain well, but PEC much less. And if you see this movie, PEC show squeezed lumen and much less drainage, but lymphatic, by contrast, they show relaxed vessel lumen upon the pressure from outside, and this relaxed behavior is correlated with this very quick and faster uptake of these red colored particles into this lymphatic lumen, which is already known and discussed in this seminar review in 2010 Cell paper from Dr. Alitalos. And this lymphatic exists like this collapse manner, but once you have pressure increase outside the vessels, these vessels become relaxed and they uptake this fluid and particles, even cells. With that, we studied some of the mechanism of lymphedema, and long story short, we found ROCK2, one of the subtype of ROCK, which is a rho associated protein kinases, is important for junction zippering under cytokine-mediated inflammation. So we tested different types of cytokine, but I just show one cytokine here. But some of the cytokine make this lymphatic junction tighten, and ROCK2 knockdown prevented this type of zippering in vitro and in vivo as well. And so we found ROCK2 has different roles in cell-cell adhesion in lymphatic. They form some unusual protein complex with GEM-A, a tight junction molecules with lymphatic endothelium to tighten this lymphatic junction under inflammation. So with that, we had some of this impact on this area. So we got these very interesting papers and feature in some of these paper cover, also some awards from NIH and NSF. And I didn't really explain this study, but another study in lab on a chip recently published, they show this paper show primary lymphatic valve dysfunction, which is quite interesting. But yeah, due to the time limitation, I couldn't really include all of them. And recently, we used the same device to understand lymphatic drug delivery on chip, because now people understand how targeting lymphatic is important and how this could be useful to regulate immunity in different types of immune disease or even in cancer. And we try this device to tell if this device could be useful to screen these different types of nanoparticle or to understand some mechanism of this delivery of nanoparticle to the lymphatic. So in this schematic, if there is a subcutaneous injection with some therapeutics, is usually the bigger size particles get into this lymphatic because of this lymphatic structure and their junctions, and the smaller one and water soluble components go into this vascular capillary. And we know these things, and we was wondering if we could study this a little bit deeper and understand something that we don't understand so far. So this is the device. The same one has lymphatic vessels, and the other channel is filled with nanoparticle with fluid. So these nanoparticles get into this lymphatic, and we measure this lymphatic drainage and understand some of this transport, which is highlighted in this ACS Biomaterials journal. To do that, we use different sizes nanoparticle or PLGA nanoparticle with 30 nanometer, 50 nanometer, and 70 nanometer, and characterize their size and their charge. And then we use the same schematic. So we had the lymphatic on the right side channels and loaded this different size of nanoparticle, which are labeled, and then got their the amount of this nanoparticle uptaken by these lymphatics. First, we just tried the device with no cells, just to understand their transport without any cell components, just the collagen bulk and the pressure gradient. And we found this trend that the smaller nanoparticle get into this right side channels and drain much quicker than the larger size nanoparticle, which is quite general, right? Because a smaller size nanoparticle move faster than the larger one. And then we added lymphatic endothelial cells to the system and compare with this acellular control group and with this LEC. And we found very striking things that in 30 nanometer nanoparticles, these the gray color is acellular, and the dark color show lymphatic endothelium included. And lymphatic endothelium, they reduced delivery of this nanoparticle, especially when the size of nanoparticle is small, which was quite striking to us. And it was also shown in 50 nanometer as well. But we didn't see that much difference in 70 nanometer PLGA. And we calculate this lymphatic resistance by dividing this total delivery with this LEC and by this acellular condition, and they got increased. So it was quite interesting. And we found when we stain these cells, DAPI and also nanoparticle in green, we found this smaller nanoparticle shows these green color nanoparticle, they are accumulated within the cytosol of these lymphatic endothelial cells compared to this bigger nanoparticle. So we thought there might be some unexpected mechanism of the cytosolic accumulation of nanoparticles in lymphatic endothelial cells. To better understand the mechanism, we use 4 different types of inhibitor, including dinosaur, nystatin, and amiloride. All these are targeting transcellular transport like endocytosis, but different mechanism, dynamine mechanism or caveolin dependent or macropinocytosis dependent mechanisms are inhibited by these 3 inhibitors. Also, we use adrenaline to target this paracellular junction-mediated delivery of these nanoparticles. And very surprisingly, when you use this dinosaur, the pink bar is dinosaur treatment and the gray bar is DMSO. And dinosaur, they increase the smaller nanoparticle uptake by lymphatic compared to the DMSO. And 50 also similarly, they increased. But the larger one didn't show any difference. When we use nystatin here, we saw the opposite. The nystatin is caveolin-dependent endocytosis inhibitor. We didn't see the difference in smaller nanoparticle, but the larger one showed dramatic reduction of this nanoparticle delivery. But amiloride, the pinocytosis shows largely variable, but we couldn't see the statistical significance. And the adrenaline, it inhibits, it delays this delivery in all the different sizes of nanoparticles. That means this junction is one of the consistent pathway of this nanoparticle delivery into this lumen. But when the nanoparticle is smaller, these cells, they capture this nanoparticle and they just keep them and delay their transport to the lymphatic lumen based on this dynamic-dependent endocytosis. And we could inhibit this endocytosis using dinosaur that increase this type of delivery. By contrast, in larger nanoparticles, they are not trapped in lymphatic, and they just transcytosis. They experience transcytosis, and they just deliver. But if you block this caveolin-dependent endocytosis, they reduce this type of delivery. So we discover using this system, there are different types of size-dependent mechanism of this delivery to the lymphatic. So if you want to target lymph node, and the size of nanoparticle is very small, I think we might need to consider this abnormal accumulation through this dynamine. But if you want to target lymphatic themselves to improve lymphatic function, I think this is pretty useful. So smaller size is better to engineer the lymphatic capillary in the periphery. So this is a new insight in this lymphatic delivery field. I want to share some of these student contribution, because many times all the research, all these things are contribution by students, but many times we just share their name. But these are my lab members. Some of them graduated recently, and they contribute to this lymphatics on a project, and they got really good fellowships and grant. And one of the students is now in postdoc in Mayo Clinic, and Samantha is working now rheumatoid arthritis and lymphatics on a chip, and Isabel, she was very exceptional undergraduate student, and she got numerous awards from the department, and also she was one of the first author of this cell and CMB paper, having 4 different flows and VEGF-C and VEGF-A. And she is now doing her MD study in Albert Einstein, covering all 4-year tuition covered by this university. And Isai and Vicky, they joined the lab recently, and they are opening some new avenue like lymphatic glycocalyx and lymphatic mural cell interactions. And our second topic is cancer, but I want to skip this part because this cancer is a little bit out of focus. But I want to highlight the impact, and we have some very interesting study in cancer metastasis, especially breast cancer, organ-specific metastasis, and also some of the studies still in process in cancer immunotherapy by modulating lymphatic function. And thanks to this funding. And these students are really excellent students, and especially Taejun was in our previous postdoc, and he's now senior engineer in Lumina. And Harry Pang is now leading the immunotherapy work in the lab. The last part is quite recent findings, and I want to share some of these findings using the rest of the time. And brain has lymphatics, and we know meningeal lymphatics is highly important. And good lymphatic is collaborating with meningeal lymphatic. And given that these lymphatic systems are highly important for many different types of brain disease, we try to make this good lymphatics on a chip. So good lymphatic is formed by endothelial cells. It is not lymphatic, it is blood endothelial cells in the brain and astrocytes. And these astrocytes polarize their echo 4, and this echo 4 help this convection of this cerebral CSF, cerebrospinal fluid from this artery to the vein, and they clear this any metabolic waste in the brain. And we use a similar model system, but different type of cells using brain endothelial cells in the channel and brain astrocyte in the bulk, and having the pressure from left to the right to mimic this convective flow. And we found this condition really maintain this aqua point for polarization. So if you see this bright field image, this is endothelium. And the bulk, we have astrocytes, and we stained them with aqua point 4 and CD31. We saw aqua point 4, they highly localized in astrocytes surrounding vasculatures, not that much in the bulk astrocytes, and they are really polarized, but they are not co-localized, means they are really co-culture condition, not really contamination or something, some other artifact. And we found using the system, aqua point 4 could be impaired, especially the polarization could be impaired under a certain type of disease conditions, such as inflammation. And in this case, we use LPS as a toxin. It is not really inflammation, but we just roughly use this LPS to mimic a part of inflammation processes. And we saw LPS really depolarize aqua point 4 in this graph. In red is aqua point 4 and green is CD31, and they are highly depolarized like this. And this is really correlated with this drainage impairment. So if you see this graph, healthy good lymphatic, they drain very well. But if you treat LPS, the drainage really reduced, which is correlated with this aqua point 4 LPS condition and aqua point 4 depolarization. And additionally, we added amyloid beta oligomer to understand some of the Alzheimer's disease pathology, and we saw similar reduction. And this is negative control without astrocytes. So that means this astrocytes polarization is critical in this fluid clearance in the brain, and this could be mimicked in the device. And also this device could show some of the pathogenesis of the neuroinflammation and also the Alzheimer's disease. And this inhibitor is alpha point 4 inhibitor, and also show this inhibition of alpha point 4 reduce the drainage in the device. This study was featured in the cover of Advanced Biology 2022. I think we don't have enough time, but this is super exciting last part of my presentation. So I will use some of the time and then share this part. So we also work on some other type of neurodegeneration in the eye. We mimic this eye lymphatic system. So in the glaucoma is type of edema in the eye. So glaucoma build up this fluid within the eye. So it increased the pressure. We call this intraocular pressure. And this pressure really damage these neural cells. This is retinal ganglion cells. And these cells transmit these stimuli from the eye to the brain. So they are the key components of this sight. But if this IOP, intraocular pressure, damage these neural cells, that might induce blindness finally. So this IOP buildup is the issue in this glaucoma, and it is known that one of the reasons is that the clearance of aqueous humor through the trabecular meshwork and Schramm's canals could be impaired in this disease. So we wanted to build up this eye-specific lymphatic machinery using the chip system. Could be similar one, but a little bit different engineering process help us build up these Schramm's canals covered by trabecular meshwork, and they drain this aqueous humor from outside. In our skip this process. And this process show this very nice cells. In red, they are endothelial cells, CD31 positive, and they are covered by these red cells, trabecular meshwork cells, and they drain this fluid from the same schematic from left to the right, right? And then we can calculate aqueous humor outflow. And we know that steroid-induced glaucoma is poorly understood, even though that steroids are widely used in the clinic to treat this type of inflammation-related condition in the eyes. But the main side effect is glaucoma. So we try to recapitulate this steroid-induced glaucoma first using on a chip system. So we treat the system with DMSO or dexamethasone type of steroid, and we saw the reduction of this flow rate, the outflow. This is a co-culture with TM and endothelium, and this is monoculture of endothelium, but it shows much lower than this co-culture. So it seems like, so we think TM cells destabilize this lymphatic endothelium, and they facilitate this fluid clearance. But if you have dexamethasone, they become highly compact like this. These are the low magnification, but this is the high magnification of this co-culture upper control group looks like this, but dexamethasone make this junction thicker than this. But interestingly, without TM cells, these cells, they form thicker junction in either condition, DMSO or dexamethasone. So if you quantify junction width and DMSO with two type of cells, they are much thinner than the other conditions. And we tested several inhibitors, and we found TGF-beta receptor inhibitor really reversed this glaucoma phenotype under dexamethasone. And this dexamethasone reduced the outflow, but SB inhibitor that are blocking this TGF-beta receptor, they normalize this. And this junction morphogenesis and widths are normalized by this TGF receptor inhibition. In vivo, we use mouse glaucoma models by applying a type of ointment of dexamethasone. They are releasing this dexamethasone long time, and this dexamethasone appointment, the treatment, this really caused this increased IOP, but SB inhibition and TGF inhibition reduced this pressure buildup, and the junction widths of Schramm's canals are increased but reduced by SB. And finally, we found this was related to VEGF-C. And one of the receptor of TGF-beta, TGF-beta receptor, ARC5, in and if you see this slide, the VEGF-C concentration reduced by dexamethasone, this is in vitro from TM cells. So TM cells secrete VEGF-C a lot, but dexamethasone reduced this VEGF-C expression, but the inhibition of TGF receptor increased this. And the flow rate in the device show similar trends. Dexamethasone reduced the flow rate, but TGF-beta receptor normalized. But surprisingly, this is VEGFR3 inhibitor, MAZE51, getting back to this disease condition, which means VEGF-C and R3 is critical to maintain the normal fluid outflow in the eye. Similarly, in the in vivo condition, we saw the similar trends, which is not published yet, but we hope to see the paper out. So this impact show a lot, but I'd like to highlight one of the collaboration with Dr. Young Hong, USC, show this CSF outflow by piezo-1 activation, and we was honored to work together using our chip system. Also, our talented undergrad student, Paul Soden, he developed this lymphatic on a chip for the first time, and he got numerous awards, and also MD student at UPenn, and covered by university and other scholarship for 4 years, tuition and living expenses. And yeah, and these are all the contribution. And finally, these are my lab members and collaborators, especially Dr. Young Hong and Gwen Randolph and Susan Kogan and Benjamin Thompson, Babak Merara and Raghu Katar and Dan Stemmer, Amanda Lund, and many other collaborators in this lymphatic community. I appreciate their help, and I anticipate a future collaboration with others as well. So I will stop here. I'm sorry I used too much time. So now it's 1:52, so we have 8 minutes left. So I will stop my sharing and try to answer some of your questions. So let me open Q&A session. Yeah, one of the question was this, are you suggesting there are 2 possible pathways to affect lymph flow in patients with primary lymphedema, and are these markers of the disease that suggest which path may be most effective? I don't know. So we are working on this lymphedema, and today I show only a part of lymphedema, that is inflammation context. I didn't cover that much about primary, but my lab also working on some of the genes to understand primary lymphedema, but it's still quite early. So my lab is primarily focused on now secondary in this lymphatic junction morphogenesis. Also, one ongoing study is lymphatic valve, that is a secondary lymphatic valves on a chip, which is in collaboration with Dr. Gwen Randolph and Dr. Rafa, and they are the experts in the valve in inflammation. So that is a part of our contribution, but clearly primary lymphedema like VEGFR3 defect or FOXC2 defect or GATA2. So many genes are very important for this field. So that could be our future investigation. And the next question is, sorry, there are many questions from, I think, patients or patient family asking about using some of these findings for their members, but it's a little bit beyond my ability. So I might respond to them by email, but not today here. All right, this was a great talk. Thank you. In reference to the VEGF-A induced sprouting being greater than VEGF-C induced sprouting under interstitial flow on a chip, Is this specific to skin lymphatics, or do you also see this effect on lymphatics from other organs? Yeah, which is very, very important question. So I believe if I could obtain these lymphatic endothelial cells from other organs, I would be very, very happy to perform all my experiment using different types of lymphatic endothelial cells. So far, we are getting these lymphatic endothelial cells from Dr. Young Hong, which is human skin lymphatic endothelium, which is great model system, but might be limiting this organ-specific lymphatic stuff. That I showed you the lymphatic, the eye glaucoma stuff as well. That is, we use dermal lymphatic plus trabecular measure from eye. So that means it is not really Schullem's canal, right? So we got some cells from collaborators, but it was quite hard to recapitulate these nice junction morphogenesis. So that is the limitation of the field. How we could get these cells from the right organs and right condition, and how we could maintain them would be very, very important contribution. And we really eager to test all these things in organ-specific manners. Another question is, could you clarify again about what these studies might mean for the treatment of Alzheimer's? I didn't quite understand what those studies suggested. All right, so the lymphatic system that we models using the chip show that these astrocytes and vascular interaction maintain this aquaporin 4. It is a water transporter, and this aquaporin 4 polarization is maintained in a healthy condition. But we saw some of these aquaporin 4, their expression is disturbed, and they are depolarized under amyloid beta oligomer treatment. So we didn't really include any treatment in this study, but we could say the device could be useful to model the disease. And potentially, if you could target aquaporin 4 depolarization, we don't know the biology yet, but could be potentially useful to reverse this fluid outflow impairment in Alzheimer's disease, Alzheimer's patients. Last question is here. Could this chip be implanted in patients with lymphedema to improve the drainage? Yeah, so we are now trying to do that using similar system but different materials. So the current system is based on this PDMS, highly stiff plastic. It is literally plastic. So we couldn't really plant this plastic to the patients. But if you could use some more biocompatible materials, then that could be useful to be implanted. For example, after surgery of this breast tumor with this lymphatic system around the tumors, it could be useful to that help this fluid drainage. But the key thing is that this device should be highly controllable. So we should induce anastomosis, the connection between the device and the lymphatic trunk, which is hard. So now we do some surgery, right? So many experts in lymphedema surgery, they do this. But as an engineer, we are now trying to how to use this device and let them being connected to the host lymphatic in the right side and right manner. Also, how they maintain their permeability, like junctions and their contractility, etc. So, which is highly futuristic, but we are now trying to do that in mouse model of lymphedema. So hopefully we could update with some new data. All right, so now it's 2 p.m. So thank you very much all for your attending this seminar. And I hope if we could keep continue this conversation and this discussion with researchers and also patients and their members and families. All right, thank you very much and have a nice day.